It's my pleasure to introduce our speaker for today, Dr. Christina Baer, who is the director of the SCOPE, the Sanderson Center for Optical Experimentation at the University of Massachusetts Medical School. Christina has an undergraduate degree in physics and English from Wellesley College, and then did her PhD in biophysics at the University of California in Berkeley, before in 2017 taking up her role as the director of the imaging facility at the UMass Medical School. And of course, in this role as director in the last 2 years, Christina really had to figure out how to tackle the limitations that come from the pandemic situation. And in the course of this, she's developed a fantastic online training resource for imaging core facilities that she'll tell us about. And we're really excited to hear and learn from you about how remote training and virtual training can happen in imaging core facilities. Please, Christina, take it away.
Thank you so much for that lovely introduction. It is such a pleasure to be here with you all today. What I am going to talk about is a little bit of a historical perspective now, believe it or not, as we're almost 2 years into this pandemic, about where we started with virtual training in the scope and where I'm hoping we're going, but I am very much looking forward to getting feedback and comments from the community and learning more about how you all are tackling this problem of virtual training too, as I go through this. And I'm hoping we'll have a really fun discussion at the end of my talk. So I want to start off especially by thanking Joanna for her patience with me via email as we've worked to coordinate me coming to speak with you today at the virtual pub. And I'm looking forward to talking about the training resources that I've been working hard to put together, really to support my imaging facility at UMass Chan. And prior to the COVID-19 pandemic, my facility conducted 100% of training in person, which I think is probably very similar to a lot of people that are on this call. And as I'll describe, I've built out the SCOPE's virtual training considerably since 2020, and I've learned an awful lot about virtual training and virtual resources as we go through that process. I'm going to mention throughout the talk several companies that we've worked with and different software packages that we've used currently in the scope. I want to make it clear that I have no financial interest in any of these companies or any of the products mentioned during this talk. These are just solutions that have worked well in the scope and in my core. So as Joanna mentioned, I'm the director of the Sanderson Center for Optical Experimentation, which we abbreviate the SCOPE. And this is the light microscopy imaging core facility at UMCMS. I have 14 light microscopes that perform a wide range of imaging techniques. In the SCOPE core facility itself, I'm very lucky to have about 3,000 square feet of lab space, which includes imaging rooms for the microscopes, wet lab space for sample preparation, a separate tissue culture room, and an animal prep room. We have about 60 annual internal users, and I have multiple external companies and academic labs from other universities that typically come to the scope to use the facilities during each year. And the facility was established in 2018. So I'm the first director of this facility in this particular iteration. So that actually gives me a fair amount of freedom to make the facility into what my vision is, which has been really helpful as I try to ensure that we have the flexibility necessary to respond to the COVID-19 pandemic and different user demands. And I'll talk a little bit about that. The SCOPE user base consists of graduate students, postdocs, and technicians primarily. So any undergraduate users come from outside institutions and are relatively rare. There's no engineering or physics department at UMass Chan. So that means that my users do not tend to try to solve technical problems with the systems by themselves. They usually come to me for help, luckily. But that also means that there's a limited amount of interest in understanding the fundamental principles underlying most advanced imaging techniques. So this is something that I've had to keep in the forefront of my mind when developing virtual training. Research at UMass Chan is primarily biology focused with a heavy emphasis on human diseases. So this short video gives you a glimpse of the main wet lab area and some of the imaging areas within the scope itself. In addition to the spaces that are shown here, we have several other small imaging rooms, dry lab space, tissue culture space, animal prep facilities, and an optics workshop. So here you'll see some of our custom live sample imaging systems, some of the optics development lab space, and our Leica SP8 confocal in use. So this probably looks fairly familiar to everybody here. I have a wide variety of instruments in the facility that cover a large range of optical approaches, from super resolution to FCS to multiphoton to a rudimentary light sheet that I built for cleared tissue to more standard confocal and wide field microscopy. When I started as the core director, we ran about 50% home-built equipment, 50% commercial equipment, and that has trended more towards the commercial equipment as my user base grows, because I personally find it much easier to support in a single staff member imaging core facility. Now, home-built instrumentation is absolutely fantastic, and it's one of my favorite things to do is to customize instruments. But because I am the only staff member supporting all of my users and all of these microscopes, the commercial systems tend to be much easier for me to get users to use initially. So I also provide access to automated imaging for histopathology sections, Marefish spatial transcriptomics, and our BSL-3 imaging instruments. And I'll talk a little bit more about those imaging solutions later on in the talk. And I strive to provide complete microscopy support, from sample preparation, protocol consultation, to microscope modification, as I mentioned, and construction, to image analysis support for our users. So at UMass Chan, there's a significant history of research groups, both using and developing advanced microscopy techniques. I want to be clear about that. But that knowledge has tended to be quite siloed in these individual groups. One of my main goals as the core director is to increase the utilization of microscopy approaches for groups across the institution through a variety of user education initiatives, increase access to advanced instrumentation through the core itself, and research collaborations with myself. The virtual training resources and different facility services that we've started to offer are specifically designed to encourage novice users to engage with microscopy techniques and incorporate those into their research going forward. So what I've developed in the scope may or may not be applicable to you based on your user base, but I wanted to provide that context before going into what exactly we're doing so you understand kind of the motivation behind what I've developed. This timeline will give you an idea of the time frame over which I developed and rolled out our initial online training offerings. So in mid-March, the university shut down for all operations except for SARS-CoV-2 research. So the scope was closed except for those researchers. We only had 2 or 3 at that time. So this was back in March 2020. I reopened at the end of May to trained users only. And we immediately implemented virtual tech support via remote desktop viewing and control software, which I'll talk about next. No trainings or in-person tech support were done at this point because we didn't know how to do it safely. By mid-July, I had a number of users asking for training. So I did some trials using Zoom and remote desktop access to essentially conduct the same sort of trainings that I had previously done in person. I don't know if any of you had this experience, but for me, this really did not go very well. So users did not retain the information over the course of a 2-hour-long Zoom session with me operating the microscope and them watching without any technical background or any previous work done on their part. It's just too much detail to absorb virtually in a long session. So as we all know, it's very different when you're not in front of the microscope and physically handling the equipment while being trained. So by mid-August, I'd started to develop the more general aspects of our virtual training, and these were rolled out in early September. And then I began developing the microscope-specific parts of our training for individual instruments. So I've been rolling those out gradually from the fall of 2020 through this current point where we're at as I complete the training modules. By the end of 2020, I'd trained 19 users remotely on a small subset of our instruments. And to date, we've had 56 users go through our current hybrid training that right now is combining virtual training and in-person sessions. So this chart shows how dramatically usage dropped off during our initial campus shutdown, when the scope was only open to previously trained users doing SARS-CoV-2 work. So coming back from this period in early June 2020, I knew the majority of users would be fairly rusty when it came to their microscopy skills. And also that we had a big backlog of projects that needed to be completed quickly, in addition to summer students trying to come in and get trained. So we had a lot of people putting pressure on the system at that point. But at this point, I wasn't approved to do any in-person training at all whatsoever. So the first virtual training pipeline was implemented pretty quickly to try to start alleviate this pressure on our training system and our resources. This floor plan shows the main part of my facility. So I also support a couple of remote instruments in different buildings, but most of my instruments are located in the space shown here. From a pandemic standpoint, we were pretty well equipped to continue operations, thanks to having the majority of our microscopes in individual instrument rooms that could be isolated. So this is one of the times when I'm grateful to have this kind of old school core facility with one instrument in each room. But none of our rooms were big enough to maintain 6 feet of distancing. So I couldn't do any in-person training with that restriction in place. I also knew that I'd need to do tech support for users on the equipment isolated in rooms all the way throughout the facility without being able to enter those rooms while they were in there with them. So remote access to all of the microscope control computers was the first thing I introduced to make sure that I could help my current trained users when they ran into issues. I considered a number of options for remote access software. And I'm sure many of you are familiar with TeamViewer and some of the functionality that Zoom has rolled out to enable screen sharing and remote control. Also, Teams is a really good option too. During the time where I was making this decision, I settled on Splashtop, which is very similar to TeamViewer, but for us, the license is cheaper. And in an imaging core, the bottom line really matters a lot. So far, this platform has worked out really well for us. I have a wide range of operating systems in the facility, as I'm sure many of you do. And this is much to the dismay of the university IT team, right? This is not something that they particularly appreciate, but we have everything from Windows XP systems all the way up to more current operating systems, Linux, Macs, you name it, right? So I needed a solution that was cross-platform compatible and that allowed me multiple layers of security control over access to the scope computers. Also, that enables communication with the users and allows me to fully control the systems remotely. So far, Splashtop has done that pretty nicely for us. This is a screenshot of the SP8 control computer with the Splashtop chat window open on the side. You can see it over here on the right. We don't have cameras or audio on all the computers or in all of our imaging rooms. And because of the way the scope is constructed in the interior of an older cinder block building, there's not cell service and our Wi-Fi can be fluky. So the text chat is how we handle communicating with users, and it's worked pretty well for us so far. Luckily, all of our systems are hardwired to the Ethernet, so we haven't had an issue with network drop on the user end. Which has helped things quite a bit. I do like that we have the option with Splashtop to save all user interactions in the chat log. So that's a good record for hourly billing, which is always a benefit. And it's also a good reference about what support was needed by each user if we need to review that. Splashtop allows me to log into any of the scope computers at any time, and I can be logged into multiple ones at the same time. Even our CentOS running Delta Vision systems, which are a little bit more of a challenge to communicate with typically using other platforms. For the Delta Vision microscopes, the user chat has to be handled a little differently. I want to be clear about that. Splashtop hasn't enabled yet the chat function for Linux systems, so I use GChat on these systems as well, but that combination works pretty well. So while I was developing these materials, and while I am developing these materials, I try to keep in mind that the facility users who are the audience for everything I produce have a range of interests and a range of backgrounds. I think of all my imaging core facility users as existing on a spectrum, right? Between I just want to push a button and get my images on one side, and I can take the microscope apart and fix it without help if I want to. So the scope users are graduate students, postdocs, and technicians who are focused on getting their images to support their other experiments. In general, the users in my core facility, and this is not necessarily true across the labs at UMass, but this is true for the core facility users. In general, they aren't particularly interested in learning all the details about the optical configuration of their equipment. So I deliberately developed my training materials with that in mind. As a trained physicist and biophysicist, that meant setting aside the fundamentals of optics, which is a little painful, I will admit, and focusing instead on exactly what people needed to know to get good data. My general user training heavily features examples of poor quality images resulting from sample preparation errors. And this is deliberate to drive home the importance of careful experimental planning, understanding the capabilities of a microscope before you start imaging, attention to sample prep, proper controls, and the usefulness of running pilot experiments, in addition to always consulting with an experienced microscopist when questions arise. So these are all major challenges that I've seen users in my facility struggle with. And I'm hoping that the virtual training resources that I've put together will help draw the user's attention to these problems and prevent them from becoming major roadblocks. I also kept in mind that the scope has very few high volume users. So this means that my users often need refreshers on how to use the microscopes. It's really not uncommon at all for months to go by in between imaging sessions. So here you'll see the number of users over 20 hours annually in comparison to those under 20 hours in fiscal year 20. So my training needed to be broken up into shorter segments that allowed the users to go back quickly to exactly where they needed a refresher when they need to find that info. When developing the training program, I first came up with a list of all things users needed to know about the facility. So what instruments and services are available in the scope, and this section turned out to be the right place to explain to people why and how the scope should be acknowledged in papers, which is always a challenge for all of us, and why that matters to the facility. And then who staffs the facility, so anything that's on the admin side is kind of in our first training series that everybody takes, all about the online calendar, how we handle data transfer, how we handle remote tech support. So I wanted a baseline level of knowledge for all users about these different services that we offer and how the core functions. And our COVID-19 SOPs are in this section too. And if you do check these out on the website, they're freely available for everybody to see. You'll notice that our COVID-19 SOPs are at this point a little bit outdated. So when that was first developed, we were really concerned about surface transmission of SARS-CoV-2. We know now that that's less of an issue. So we were covering a lot of things in the microscope room with plastic wrap. There's so much plastic wrap in those first SOP videos. So I do still train people using those videos, but when I bring them into the facility for the hybrid live in-person part now, I tell them we've stepped back a little bit and these are the current precautions that we're using. And then finally, each user needs to be trained to use different microscopes depending on their project. As I mentioned, I've got 14 light microscopes in my core facility. Practically every single one of them comes from a different manufacturer. And so universal training is not going to address questions about all of them. I can't even have one video about turning them all on because they're all different, right? So that was a list of what all users need to know, but there's also a list that we all have in our heads, right, of what users should know. So I decided that I wanted to highlight several areas, including how to correctly design an imaging experiment. Also, a basic understanding of the advantages and disadvantages of their imaging approach. Anyone using a confocal should, at minimum, know why they are using a confocal instead of a wide field microscope, for example. They'll also need to know what information is essential to include when writing their methods section. And finally, all users should know how to evaluate imaging data quality. And this is actually something that I feel pretty strongly that all scientists should know how to do, but we're going to start with my user base and see what I can do in terms of increasing their education about this issue. So this is something that no matter how much microscopy my users do going forward in their scientific careers, they're going to be evaluating other people's microscopy data in papers they review. So having an understanding of what makes a good image and what makes a quality image for publication is really critical. And it's something that I don't know that we, at least at UMass, were necessarily teaching very well. And I don't know that I've necessarily figured this out either, but it is one of the motivations for this training approach. So what does my training workflow look like currently? There are separate sections that each have several video modules associated with them. The first section covers the important information about the facility, the need to knows, and the second covers important microscopy basics, the should knows. And then the user moves to microscope specific training that's split into basic and advanced operational modes. So all of this training is done on our website as shown here. So you can find that all and everybody's free to take a look at it and check it out. There are 6 videos with quizzes at the end for the general basic training. And those quizzes help me track who's completed the training and also confirm that the users watch the videos. They're not particularly challenging quizzes, but it does help with the logging and compliance. And then this is an example of microscope specific training. There are 8 basic video modules for the Leica SP8 shown here, and then 6 additional advanced modules. Because not all users will need to use all of the advanced modules, I don't require quizzes at the end of those, just at the basic modules that I think everybody needs to complete. So then the virtual training is followed up with live imaging sessions using the user samples. Importantly, we don't train using a generic or stock sample. I always train using the user samples because we see such a wide variety of samples in the facility that I found that to be the most effective. When I first developed this training, the live sessions were done on Zoom and Splashtop with the user samples. And this is a screenshot showing me doing the Zoom portion of the training. So we'd first do a couple-hour session with me operating the microscope while the user watched remotely. And then we'd later do a second session with the user on the system and me watching remotely. So there are some tricks to doing live trainings via Zoom, which I'm happy to discuss later if people are interested, but we thankfully have been able to return to in-person training over the past few months. That has really made this process a lot easier for me and the users, because there's absolutely no substitute for getting your hands on the microscope, even if you can drive it remotely, which everyone could do and did get a chance to do during these sessions. It's really hard to teach color illumination or focusing even on a system remotely. Doable, but kind of painful for everybody involved. So how do these modules go together? There are 3 different types included in the training. Some modules are narrated videos that include entirely original content from me. These are mostly specific to my facility and the more unique instruments that I have, so some of my home-built equipment. Then there are presentations that utilize existing video content. So I heavily used Jennifer Waters' microcourse videos, with acknowledgement, of course. And I also use some videos from microscope manufacturers too, if available. So this really helped cut down on the total time that I had to spend putting all of this information together. This is a pretty overwhelming task to put together online training. And so having access to quality training resources that you could incorporate into your own course and your own approaches really, really helps. It's important to note too that changing the narrator and the presentation style periodically will also help with the knowledge retention. So I try to incorporate as many different materials as I can when I'm putting together these short videos. And it also helps, as I just mentioned, save time because it's faster than creating my own content. So then finally, there are some microscope operations that are just easiest to show in a live format. So there I simply record videos while narrating like I would do an in-person training. And most often we're not doing this with any sort of fancy equipment. I'm often recording these with my iPhone, for better or for worse. Sometimes the video quality and audio quality is not the best, but it seems to get the job done. So there are advantages to virtual training, and I will be continuing with it going forward in the scope. And I'm working to build out the resources that we've got for training on the website. So I want to note here that the training pathways on the website are still in development. So please check back if you go there looking for something that hasn't been posted yet, or shoot me an email and just ask when it's going to be posted, because this is something that I have to do whenever I can squeeze in the time to make these videos. So tailoring the content to my users rather than pointing them toward more general overviews about microscopy will hopefully help with the retention of critical information. And it also gives them a known resource to return to and gets them used to listening to my voice and coming to me to get answers to their questions rather than just going to Google, which has its pluses and minuses. It's fine if they want to do that. But I do like to encourage them to ask questions directly of me so that we can talk about answers that fit within the context of the facility and the equipment that we have there. So being able to track user training and the dates of completion via quiz submission has been really helpful for us, even though it's an additional level of administrative paperwork that we need to keep track of. It's not something that we had been doing prior to the pandemic, necessarily tracking in this detail the user training. I've also helped that this process too helps to identify users that need more assistance early on. For example, if they have trouble following a training procedures detailed on the website, chances are they're going to need more help with the microscopes and trying to image independently. So this is a good early flag for me. My experienced users that only utilize the facility periodically are actually finding the training videos to be quite helpful resources. So believe it or not, they started to check the website first before contacting me for help. And that saves me time and everybody time if they can find the answers really quickly themselves. So since I am the sole person running a large facility, any resources that help facilitate these daily operations are super valuable to me. So what's next with training, right? Ultimately, I'm planning to incorporate an additional module or modules about each imaging approach that the users will complete before doing the instrument-specific training. So this is going to be like the confocal module, right? What is confocal microscopy? Why am I doing it? What are the advantages and what's the instrumentation that we have available in the scope that can be used for confocal, as one example. So hopefully that would give people a little bit better introduction to the types of instrumentation that we have, so they're aware of it for their next times when they need to use a microscope and they're not quite sure which one they want to use, since we do have a variety of instrumentation available. So the training approach that I've detailed here so far really focuses on user-operated microscopes. For several years, I've been working to diversify the business model of the scope to ensure a more reliable revenue stream for the facility. As you saw, we have a large number of users that have less than 20 hours per year annually of a use, which is a super inconsistent revenue stream, right? So what I'm trying to do is both get users to incorporate imaging more substantially into their research platforms, but also come to rely on us for quick and easy image data generation and analysis. So this year I've introduced 3 new microscopes, our tissue-gnostic slide scanning systems and our VisGen Alpha Marefish microscope, which are operated as imaging services. So that means that samples are dropped off at the facility and I do the imaging on these systems for the users for a fee. By introducing these services, I've been working to provide researchers with the opportunity to get high quality imaging data without investing a significant amount of time learning to use the microscope, right? So we obviously would all like them to spend time learning to use the microscopes, but in a lot of cases, that's just not practical. So user education, though, it turns out to still be really important here. My next initiatives are going to be focused on developing the materials necessary to ensure that imaging service users understand a few things about the microscopes, even if they aren't actually using the systems themselves. So to plan this next round of training materials, I return to the questions I asked originally. What do imaging service users need to know? And what should they know to get good data? There are some differences here from the original training approach, right? And I'm still thinking about the most effective teaching approaches to convey this information to this different user base. And I'm really curious to hear how other people are doing this if you do offer imaging services too. These are the 3 automated imaging platforms that we've got that enable whole slide imaging and spatial transcriptomics currently in the core. These systems are completely different from anything we've had in the core previously. So I've had to do a considerable amount of user education about these system capabilities, as well as advertising to get the word out that we've got this new instrumentation. So COVID has made education and advertising particularly challenging, as we all know, because we aren't able to do larger scale in-person workshops or gatherings of users like I used to do to welcome new instrumentation to the facility, right? So while a very basic imaging approach for most of us, right, tiled color imaging, prior to the introduction of the TissueFax SL in the scope, there was no automated high throughput imaging system capable of this sort of imaging at UMass Chan in any core facility. So the researchers were either using systems with an automated stage, 1 slide at a time, or surprisingly often, believe it or not, they were using manual stages still in 2018 when I started and hand tiling their image capture, right? This is incredibly time consuming and really not something that people need to be doing right now. We can automate this really easily. So having this capability is a big deal for us right now. Similarly, limited whole slide imaging for immunofluorescence samples was available too. So users are just beginning to see what these systems can do, which is a great point for me to start to build the training into what the users are doing before they drop samples off at the facility. So I've got a number of researchers asking to more highly multiplex their IF experiments. So when purchasing the tissue-gnostic systems, I deliberately loaded these scanners up with different filter sets, including the TissueFax Chroma configuration, which allows us to image the opal dyes. And I'm really excited about this configuration. We're getting gorgeous data. I mention this capability because this is an additional area of education for my users where I need to develop virtual training of teaching people about dye selection, appropriate controls, etc. So to try to make dye selection a little easier, I've added custom spectra viewers from FPBase to the website for each scanning system. But I'm finding that the users need help with these tools. I can't just point them to the website and say, here, check your dyes, and they'll figure it out, right? Particularly when we've got multi-pass filters, things like that going on. People look at this and say, I don't know what's happening here. I don't understand. So this is one of the topics that I need to address first in my next round of virtual training development. I often refer to the tissue-gnostic systems as slide scanners, but we are using them for a number of applications. So this image is from a 24-well cover slip bottom plate that has a mixture of cell populations that have also been infected with fluorescent bacteria. So we do a lot of this whole multi-well plate imaging, whole cover slip imaging too, as a substitute for flow cytometry. Imaging these cover slips allows this group to do proximity analysis for different cell types and quantify that as a function of bacterial load in infected cells. So that sort of data analysis is shown here as I scroll through these slides. So this particular project was done by Russell Ulbricht from TissueGnostics USA using the StrataQuest image analysis software package. And this is the original data. And as I click through, you'll see nuclei detection, bacteria detection, secondary cell type detection, and then finally the proximity perimeter mapping with the detected features. So this sort of analysis is frequently requested by my users, but it's very custom, right? And so given the complexity of the analysis, it's challenging to train the users to be fully fluent with the software, particularly when they often have a one-off project that they want to complete and then move along with their other experimentation rather than developing extensive expertise with the imaging software itself. So this is how I'm thinking that we'll work to implement user training for the scope imaging services. We'll start with the basics that will be essential for everyone, including our color imagers who are just looking to scan H&E stain sections. Users planning to do IF will do further training, and those interested in image analysis will have the option of additional training if they wish. We will also provide support for individual image analysis projects as a one-off service too, which is something we do right now. Currently, the way I do this is I develop a analysis pipeline for the users or a workflow, and I write them a manual that's click by click, and then they implement it in their own labs or remotely on the scope workstations using software that we have a license for. Now, this has worked somewhat. Sometimes it works, sometimes it doesn't. It really depends on the user. It really depends on their willingness to read a manual. And it turns out reading a manual is not something people are doing a lot of anymore, right? So while this written documentation is really, really great for passing knowledge along potentially to other users who are not as familiar with the software, we're finding that videos are probably going to be the better way to go in terms of narrating through the image analysis and explaining the different steps and showing what happens as you click through. It just seems to be a format that more people are used to at this point. So that's something that's a work in progress. Now you'll notice the asterisks here. This is all in development right now. So image analysis training is something we haven't talked about yet, right? So it's an entirely different challenge than microscopy training. While there are some pretty clear goals for this sort of training, there are also significant challenges that need to be addressed, right? I'm really not sure what the most effective approach will be here. So I'm really interested to hear what other people think, what they've tried, what's worked and what's not worked when it comes to image analysis. For us, much of the image analysis is project specific, as I just mentioned. So that really complicates making general training materials. So not too many users are actually interested in developing independence when it comes to image analysis software itself. They just want independence when it comes to doing their own project-specific analysis. And then they're done, right? So the fluency with image analysis software requires practice and usually requires applying that software to a number of different projects, right? And that's something we're doing in the core, but it's not something my users are doing typically. So that's something that I'm trying to keep in mind as we consider what resources would be most useful for this part of the training. With this topic in particular, I'm still figuring out which goals are aspirational versus practical and how to most effectively solve the issue of user education, while also ensuring there's a transfer of knowledge within labs to maintain the skills necessary to do image analysis in the future as graduate students leave and postdocs leave, right? We've all seen this, right? The postdocs writes code, nobody else can get it to run. The image analysis is done, and then someone needs to recreate it for a paper to make one more figure, and everybody's completely at a loss, and you can't get a hold of that postdoc for whatever reason, right? So this is both a documentation issue, but it's also a transfer of knowledge issue. And so how we maintain that is something that I'm still thinking through. And like I said, I'm eager to talk to everybody about. So finally, I wanted to touch briefly on the newest scope imaging service, which is Marefish Spatial Transcriptomics. So this is an example of a very advanced complex imaging technique that will be offered completely as a service due to the intricacies of the approach and the cost of running the samples. We won't be training users on how to do Marefish, but we will be working to have the training videos available to help them understand the approach, the background behind it, and to assist and help them with initial experiment design and the final data analysis steps. So kind of the beginning and the end of the experiments themselves. So this is a different user education approach and user education problem than I've discussed previously too, right? So Marefish stands for multiplexed error robust fluorescence in situ hybridization. That's a mouthful. Marefish is one of many spatial transcriptomics approaches that have been developed. Everybody's probably familiar with RNA-Scope, sometimes SlideSeq, a lot of the other approaches that are out there. There have been over 20, I think, that have been developed within the last 10 years. So this is a fast growing field. This particular technique, Marefish, involves a sequential labeling of hundreds to thousands of targets in intact tissue at subcellular resolution. So the detection sensitivity of this particular approach is really remarkable, going down to about 0.1 copies per cell. And it's really important from an imaging standpoint that we're getting subcellular resolution. Our initial goal for the pilot project at UMass Chan was to spatially map pathogenic cells across the brain and characterize different cell types by gene expression in different inflammatory conditions. Marefish generates about 2.5 terabytes of raw data per tissue section. That's in 24 hours. So in addition to imaging equipment, I've worked to establish the computer resources necessary to handle this absolute tidal wave of data that we've got coming in now. So this is not a solo effort. This establishing Marefish has taken a team, and this was established as part of a scientific collaboration initially between me and Dr. Dory Schaefer, shown here. And we're doing this with the help and support of two very talented postdocs in the lab, Travis Faust and Violetta Duran-Laforet. We've been really, really fortunate to have them. The funding for this came from the Massachusetts Life Sciences Center, which is a state entity in Massachusetts that provides funding to drive innovation and industry academic collaborations. And this grant funded the purchase of a VisGen alpha microscope. The scope is one of only a few labs in the world that has an alpha instrument right now. VisGen's a rapidly growing startup, and we've worked really closely with their team members shown here to get this instrumentation and the Marefish technique running at UMass Chan. So I wanted to briefly, because this is a relatively new technique, go through how Marefish works. Encoding probes are designed that are specific to each target gene in the tissue. So these probes have a gene-specific target sequence and 2 flanking sequences. The fluorescent readout probes bind these flanking sequences. Each gene has 96 different encoding probe sequences that are designed in combination with 2 flanking region sequences. So this leads to 192 encoding probes per gene. And luckily, VisGen's library design team takes the lead on probe design after the investigators supply a list of genes that they want to target. Typical library sizes are about 400 genes. And the library team does the design using existing single-cell RNA-seq data sets and then runs quality control checks before having the library synthesized. And the initial hybridization step for the encoding probes is done before the sample is loaded on the microscope. Once the encoding probes are hybridized, the remaining reactions are all automated on the microscope. The sample is loaded, and a series of pumps control the hybridization shown here of the fluorescent readout probes and then the washing of the tissue and the bleaching step. The first readouts are shown here with an example data image. So this is classic single molecule fish imaging. So it's not particularly challenging from an imaging standpoint. The output data itself isn't that complicated. It's just that there's a lot of it and we have to decode it to find the individual locations of each transcript, and I'll talk about that. So multiple rounds of hybridization continue, resulting in multiple images for each field of view. In the end, we end up with many images per field of view, the instrument's taking Z-stacks for each staining round. So that's where this massive data generation happens. We've got multi-channels going on, and we've got Z-stacks with multiple steps going on. So each of these images is then decoded using an image processing and analysis program to reveal the gene-specific barcode that tells us exactly where expression is happening. The barcodes are where the error-robust part of marefish comes in. So each target gene is assigned a 16-digit binary barcode. These barcodes consist of ones and zeros, where ones correspond to a fluorescent spot in the image and 0 corresponds to no signal. So each image from each hybridization round is decoded pixel by pixel to uncover the barcode at that location. The marefish barcodes are designed to be error-robust in that each barcode differs from all other possible barcodes by 4 changes. So it would take 4 misreads or errors in the output to cause one barcode to get changed into another, causing misidentification of the gene. So if the decoding program finds a barcode that does not match by 1 digit, we know it's a misread, and that barcode can be assigned to the proper sequence, revealing the gene ID. So this process of resolving errors continues with double and triple error detection. So this gives us very high confidence that the detected signals are real and are assigned the correct gene ID. So I've described now in detail how this works. The question is, can we get it to work in our hands? And the answer is yes. Here's some data. So this is a single field of view from a brain slice. And what you can see are the detected transcripts represented with spots here, and the cell boundaries highlighted with a dashed circle. So we can assign cell types based on this expression data as shown on the right, which was really exciting. So using some established single-cell RNA sequence analysis pipelines and some newer software, we can move from UMAP plots to spatial data very easily. So this data is, again, from Dory Schaefer's lab, and the cells here are colored by class. So you can see that not only are we identifying cell types, but we're able to identify cell subtypes here. So not just neurons, but different types of neurons based on the expression profile of each cell. This really gives us an unmatched view of what's going on in brains under different conditions and in different organs too. So we are seeing spatially organized expression patterns, which we expect in a highly organized organ like the brain. We have run marefish on olfactory bulbs and on brain tissue so far from both mice and humans. And we're working on expanding into other organs as well, such as lungs and skin. So in the scope, we're working with labs to do marefish, and we now have a well-established workflow for library design, sample quality control, sample preparation, and data processing and analysis. So thanks to the funding from the Mass Life Sciences Center, we have all the hardware available for both the imaging and the analysis. So supporting this workflow and the instrumentation have required us to build out these resources to make this technique possible. So I wanted to show you this as an example of how we're handling marefish from the core side to make it feasible for our users. The experience of adapting our imaging facilities and course to training during COVID has provided us all, I think, with a testing ground for new education approaches, some of which I've just detailed for you, that will enhance and strengthen global microscopy knowledge across our user base and hopefully across scientists as a whole. I do want to highlight the breadth of data types that are captured in my core facility here with these 2 videos. And I know that the scope is far from unique in this regard. Imaging core facilities offer this unique concentration of knowledge in one facility and in the staff members themselves. So moving to online training is giving us this fantastic platform for capturing and sharing the depth of that knowledge in a more accessible manner. I wanted to end today by opening up the discussion about what virtual training resources will look like over the next 5 years. So based on my experience in my own facility and from the feedback that we've gotten to the BINA, Bioimaging North America Training and Education Working Group, there's a broad need for training materials across all levels. The way that I see this as we have this huge mission right now to develop resources that are applicable for users all the way from universal to expert training resources. A question for the imaging communities as a whole is going to be how we prioritize the development of these materials. We're all very busy in our own core facilities and we're all short-staffed, right? So how do we come together and to support the development of these training materials in ways that are both applicable for your own individual core facility, but also reflect the values and priorities of the global imaging community as a whole? And then how do we support these efforts to develop these resources that are going to benefit all of us in the long run? In addition, there's questions about how we identify knowledge gaps. What are the priorities? What are the targets? This is going to be different for every imaging core facility, and everybody's going to have a different opinion about that. But I'm hoping that there's going to be enough common ground that maybe we can share some of this workload, because developing these training resources does take significant time and planning too. So finally, I want to briefly acknowledge people who have been involved in some of this work. So at UMass, Dorothy Schaefer is my main collaborator on Marefish, and we're working to bring Marefish to all of the labs at UMass and external labs too. And this wouldn't have been possible without help from Jeff Moffitt at Boston Children's, who was one of the inventors of the Marefish technique. The SCOPE core facility itself operates under my direction, but with the guidance of a very experienced scientific advisory board, whose names are listed here. And then the experimental approaches in terms of the imaging services that we offer would not be possible without equipment and technical support from TissueGnostics and from VizGen. And I've been very grateful for all of the assistance that both of these companies have provided over the past couple of years. And I'm really looking forward to partnering with them in the future to continue to expand the training resources and the instrumentation that's available for my users. And then funding for all of this comes internally from UMass, but then also from the Massachusetts Life Sciences Center. So I'm going to flip it back to this slide, and I'm going to open it up for any questions. Thank you all so much for hanging in with me as I worked through all of this.